Okay, let's unpack this. We're diving deep today into something pretty fascinating and honestly sometimes quite heartbreaking in genetics.
Hmm.
It's this puzzle. How can two parents, completely healthy, no signs of any disorder, end up having more than one child with the exact same, often severe, autosomal dominant condition?
Yeah, that paradox, it really seems to fly in the face of everything we sort of learn about basic inheritance, doesn't it?
Totally. It
And,
feels like it breaks the rules.
and that paradox is really the core of our deep dive today. It points us towards this specific kind of hidden mechanism called gonadal mosaicism.
Gonadal Mosaicism, okay.
Yeah, it falls under this umbrella of single gene disorders, but with what we call non-classic inheritance. Basically, the simple Mendelian rules we learned in school, they don't quite cover the situation.
So the mission today is to get into the weeds on this, understand the mechanics.
Exactly. Where does this stealth mutation actually live? Why do the parents seem perfectly fine? And critically, what does this mean for the risk? The risk of it happening again in their family? Because that risk is often missed by standard approaches.
Right, and this isn't just like a theoretical thing for geneticists arguing in a lab.
Oh, absolutely not.
Understanding gonadal mosaicism is crucial for genetic counseling for families who are actually facing these situations with diseases like osteogenesis imperfecta, for instance.
Definitely. And really, for anyone listening who wants to grasp these subtle complexities of how DNA errors can pop up and get passed down in unexpected ways.
Okay, so here's where it starts getting really interesting, because the timing seems to be absolutely critical here.
Timing is everything in this story.
So let's start with the basics, just so we really appreciate what rule is being broken. When we
Yeah.
talk about a typical autosomal dominant disorder, what's the standard expectation? Especially when it shows up seemingly out of the blue in a family.
Okay, so the standard rulebook says, if a child has an autosomal dominant disorder, meaning, just one copy of the bad gene causes the illness, they usually inherit it from a parent who also has the condition.
Because it's dominant. You see it if you have it.
Exactly. Now, the main exception we normally think about is a de novo mutation, a new mutation, spontaneous.
Meaning it just happened randomly in that specific child. It wasn't passed down.
Precisely. In that standard de novo picture, the mutation occurred spontaneously in the single sperm or the single egg cell that happened to form that child. It's seen as a one-off biological glitch.
A bit of bad luck in that one conception event.
Right. And because neither parent carries that mutation in their body cells, they're what we call constitutional DNA,
Yeah.
and the error was unique to that one gamete. Well, the recurrence risk for future siblings is considered basically negligible, less than 1%, like lightning striking twice.
So the usual counseling message there is pretty reassuring, right? This is just a random chance event, very unlikely to happen again.
That's the standard message. But, and this is the big but, in the cases that point us to gonadal mosaicism, that comforting idea just falls apart.
Because you see it happen again.
You see it happen again. Multiple affected children born to parents who are phenotypically, outwardly, completely normal. That pattern of recurrence, seeing the same dominant disease pop up 2, 3, maybe more times, that clearly violates the laws of Mendelian inheritance.
Because it's not random anymore.
It implies a stable, repeatable source of the mutation. It tells you something is fixed, something internal, recurring within one of the parent's reproductive systems. It can't just be independent, random chance each time.
And you mentioned osteogenesis imperfecta, OI, as a classic example here. That's the brittle bone disease, right?
Yes, exactly. OI is often used because it's typically caused by dominant mutations in collagen genes like COL1A1 or COL1A2, leading to really fragile bones. And it's usually highly penetrant. If you have the mutation, you have the disease, often severely.
So it's usually obvious if a parent has it.
Correct. So imagine a couple. They have one child with severe OI. Tests show it's from a specific known OI mutation. But neither parent has any sign of brittle bones, no symptoms at all. Initially, everyone says, okay, de novo mutation.
Standard explanation.
But then they have a second child, and that child has the exact same severe OI mutation. That's the red flag. That's the moment alarm bells go off.
Because the odds of the exact same rare mutation happening spontaneously twice in the same family are astronomically low.
Vanishingly small. The fact that parents who seem perfectly healthy can have 2 or more children with this specific severe condition is the key evidence that forces clinicians to look beyond the simple de novo model and consider this hidden mechanism, gonadal mosaicism.
It's not just bad luck anymore. It's a pattern demanding a biological explanation. It was seeing this recurrence in OI and other conditions too, despite the parents testing negative in their blood, that really solidified GM as a recognized clinical phenomenon.
Absolutely. It showed there had to be a reservoir of the mutation somewhere.
Okay, so if we accept there's this hidden stable risk inside one of the parents, how does the mutation actually hide itself? How can it be present in their reproductive cells but not affect their own body?
Right. That's the million-dollar question. And the answer, as you hinted earlier, lies in the timing of when the genetic mistake happens.
You said timing is key.
It really is. Gonadal mosaicism stems from a new mutation that occurs postzygotically. Let's break that down. Post meaning after. Zygote meaning the fertilized egg. So the mutation happens after fertilization.
So wait, when the parent themselves was conceived, the initial cell, the zygote formed from their mom's egg and dad's sperm, was actually genetically normal.
That's the crucial point, yes. The error wasn't inherited from their parents. It happened within them during their own very early embryonic development.
Wow. Okay, so as that fertilized egg started dividing, becoming 2 cells, then 4, then 8.
Exactly. Somewhere in those very early cell divisions, the mistake occurs in one cell. Think of it like photocopying. You start with a perfect original, the zygote. You make copies. Most copies are perfect. But one copy machine makes a typo early on. Every copy made from that faulty copy will carry the typo.
So 2 cell lines start developing side by side in the embryo that will become the parent. One line is normal, the other carries the mutation.
Precisely. This leads to the state we call mosaicism. The individual is built from genetically distinct cell populations. But here's the critical part for gonadal mosaicism. Where do those mutated cells end up?
Okay.
Due to the timing and the way embryos develop, the cell lineage carrying that mutation gets sort of channeled or restricted. It predominantly, or sometimes exclusively, contributes to the cells destined to form the gonads, the testes in males, or the ovaries in females.
The reproductive organs.
The reproductive organs which contain the germ cells, the sperm and egg precursors. So the mutation becomes localized, trapped, if you will, within the tissue that makes the gametes.
Okay, let's really spell out these 2 different cell populations existing inside that parent, because that separation is key to the whole paradox.
Absolutely. First, you have the vast majority of cells.
Yeah.
The somatic cells. These are everything else. Skin, blood, bone, brain, muscle, you name it. In a parent with gonadal mosaicism, these somatic cells are derived almost entirely from the normal cell line. They don't have the mutation.
Which is why the parent is phenotypically normal. They look healthy, feel healthy, their bones aren't brittle, etc.
Exactly. They are, in effect, biologically shielded from the mutation's effects because it's not in the tissues that make up their body.
Okay, so that's population 1, normal body. What's population 2?
Population 2 resides within the gonads, the germ cells, or more accurately, the germline stem cells. A specific population of these progenitor cells, the ones that will keep producing sperm or eggs throughout the parent's life, do carry the disease-causing mutation. It's like a patch of mutated cells within the gonad.
A stable patch.
A stable patch.
Yeah.
It's established early, and it persists, continuously generating gametes, some of which will carry the mutation.
So the parent is a mosaic, yes, but it's a geographically isolated mosaicism. The mutation is locked away in the reproductive tissue, essentially. The healthy body acts as the perfect disguise.
That's a great way to put it. The disguise hides the recurring reproductive risk. #tag Nancy. Implications for offspring and recurrence risk.
Okay, now we understand the setup. Healthy parent, but a hidden pocket of mutation in the gonads. What does this internal geography mean for the chances of passing the disorder on?
It means that every time that parent conceives, they're essentially drawing from a mixed bag of gametes. Some sperm or eggs are normal, derived from the healthy germline cells, but some carry that dominant mutation derived from that mosaic patch.
So it's not like a single random accident anymore. It's like drawing from a lottery barrel where, some specific percentage of the tickets are, well, bad tickets.
Exactly. It's a rigged lottery to some extent. And this brings up the really practical but often difficult to answer question. What's the actual risk percentage? The source material rightly emphasizes that the recurrence risk depends entirely on the proportion of those germ cells that carry the mutation.
That proportion is the key variable.
It is. If only, say, 1% of the sperm or eggs carry the mutation, the risk per pregnancy is 1 in 100. If it's 10%.
And it's a 1 in 10 chance every single time.
Right, let's compare that. Standard dominant condition, affected parent, 50% risk, 1 in 2. Standard de novo mutation, less than 1% risk, maybe close to 0. Gonadal mosaicism, it sits somewhere in between, but significantly higher than the de novo risk.
And potentially much higher. What do studies actually show for that proportion?
Studies looking at families where GM is confirmed, especially in conditions like OI or Duchenne muscular dystrophy, which is X-linked but can also involve maternal GM, suggest that germline mosaic proportion could vary quite a bit. It might be as low as 1 or 2%, but it could definitely reach 10%, 15%, maybe even up to 20% in some documented cases.
Wow, so a 1 in 5 risk in some cases, even though the parent shows absolutely no signs.
That's the potential reality. Think about what a 15% risk means. That's roughly a 1 in 7 chance with each pregnancy. That is a massive, life-altering risk that the family would be completely unaware of if they only looked at the parent's health or relied on standard blood tests.
And that is the link back to the paradox, the why, behind multiple affected kids.
Exactly. The stability of that mutated patch of germ cells explains the recurrence. The mutation isn't a fluke each time. It's a constant feature within that parent's reproductive system, making recurrence a predictable statistical probability. The tragedy is that often the only way this risk is revealed is after a second or even third affected child is born.
So functionally, for that specific gene and that specific parent, their personal mutation rate is way higher than the general population rate. It's like a localized genetic fault line.
That's a perfect analogy, a localized fault line. And the higher end of that risk spectrum, that 15 or 20%, is why, tragically, some families experience this devastating recurrence multiple times before the diagnosis of Gonadal Mosaicism is finally considered and confirmed. It shifts their understanding from unbelievably bad luck to a defined biological mechanism. #Tag2. In-depth exploration of clinical and diagnostic implications. #Tag A. Classification is non-classic inheritance.
Okay, so we've established GM breaks the standard rules. It's specifically called one of the types of single gene disorders with non-classic inheritance. Why is that classification important? What does putting it in that category alongside things like, say, triplet repeat disorders or genomic imprinting tell us?
That classification is really important because it highlights why the standard rules don't apply. Classic Mendelian inheritance operates on the assumption that an individual's genome is the same in all their cells, what we call a constitutional genome.
One blueprint for the whole body.
Right. Non-classic inheritance mechanisms, like GM, trinucleotide repeats, imprinting, mitochondrial inheritance, they all introduce exceptions to that rule, or they alter how genes function in ways Mendel couldn't have predicted.
So for GM, the exception is about location. The genome isn't uniform across the body versus the germ cells.
Exactly. It's a spatial separation. Gonadal Mosaicism fundamentally demonstrates that functional genetic differences can exist between the somatic genome, the body, and the germline genome, the reproductive potential, because of events happening after fertilization. It breaks that assumption of uniformity.
How does this differ from other kinds of mosaicism we sometimes hear about? Like I've heard of mosaic Down syndrome, where someone has milder features because only some of their body cells have the extra chromosome. That's also mosaicism, right?
It is, but it's a critically different type. That's somatic mosaicism. Like GM, it arises from a postzygotic error. But in somatic mosaicism, the mutated cell line contributes significantly to the body's tissues, skin, blood, brain, etc.
So the person is affected, maybe just less severely, and you can detect it in their blood or skin.
Precisely. The individual usually shows a variable phenotype, often milder than the full non-mosaic condition, but they are clearly affected, and standard tests on somatic cells, like blood, will detect the mosaicism.
But with GM.
With GM, you have that biological insulation we talked about. The cell line carrying the mutation for, say, OI or Duchenne is restricted only to the gonads. The parent somatic cells are mutation-free. That's why they appear normal, why standard tests fail, and why the risk is hidden until transmission occurs. It's clinically invisible in the parent.
It really is the ultimate stealth carrier state. Constitutionally normal, but reproductively carrying a specific risk. #B, the diagnostic challenge.
And that invisibility leads straight into the biggest diagnostic headache. Think about the clinical workflow. A child is diagnosed with a clear autosomal dominant disorder. Standard procedure. Test the child to confirm the mutation, then test the parent's DNA. Usually from a blood sample. What happens in a GM scenario?
The parent's blood tests come back. Completely normal. Negative.
Bingo. The lab report will say, mutation not detected in mother, or father does not carry the mutation. Because, of course, the mutation isn't significantly present in their blood cells, which represent their somatic genome.
So the standard test fails to see the risk.
It fails completely. And that's the core diagnostic challenge. The parent is constitutionally normal in all the easily accessible tissues we typically test. So how do you diagnose GM?
Well, you mentioned the tragic way after a second affected child is born. That pattern becomes the diagnosis, retrospectively.
That's often how it happens, yes. The recurrence is the primary evidence. The only other definitive way would be to directly test gonadal tissue, do a testicular or ovarian biopsy.
Which sounds invasive and not really practical or ethically straightforward.
Exactly. It's rarely, if ever, done just for diagnostic confirmation of GM. So for many families, the diagnosis remains presumptive, based on the recurrence pattern. However, there's a potential technological angle here with newer sequencing methods.
Ah, okay. So standard tests miss it. What about more sensitive tests?
Right. Standard sequencing methods, like Sanger sequencing or even routine next-generation sequencing NGS on blood, usually need the mutation to be present in a decent fraction of the cells, maybe 5–10% or more, to reliably pick it up.
But in GM, the mutation is almost exclusively in the gonads, not the blood.
Mostly, yes. But because the mosaicism arose very early in embryonic development, there's a small chance that a tiny fraction of cells derived from that mutated lineage might have ended up in the bloodstream or other somatic tissues. Maybe due to cell migration during development, maybe the separation wasn't 100% perfect. We're talking maybe 0.1%, 0.5%, a very low level.
Too low for standard tests.
Too low for standard tests. But modern deep sequencing technologies, or highly sensitive mutation detection methods, can sometimes pick up mutations present at very low frequencies, even below 1%.
So the hope is that even if 99.9% of the parent's blood cells are normal, a super-sensitive test might detect that 0.1% carrying the mutation.
Exactly. Finding even a very low level of the mutation in a blood or saliva sample provides strong, non-invasive evidence supporting a GM diagnosis. It confirms the parent is mosaic, even if the level in their blood is far too low to cause them any health problems. It validates the recurrence risk.
That's a really crucial point. The diagnostic failure isn't just about the mutation being in the wrong place, gonads versus blood. It's also about the amount of mutation present falling below the detection threshold of standard tests. Deep sequencing pushes that threshold lower.
Precisely. But it's not guaranteed to find it, and these tests can be specialized and expensive. So often, even with advanced tech, the diagnosis still rests primarily on that clinical pattern of recurrence. This forces genetic counselors to work with risk estimates rather than definitive genetic proof from the parent themselves. #TaxTerces counseling and risk management.
Okay, let's talk about counseling. Knowing it's Gonadal Mosaicism, not just a random de novo event, must completely change the conversation with the family.
Oh, absolutely. It shifts everything. The emotional impact is huge, moving from lightning struck twice to there's a persistent internal risk. The first step is explaining clearly why the initial de novo label was likely incorrect, and that one parent is, unknowingly, a stable carrier in their germline.
And then you have to talk about the recurrence risk, which you said could be anywhere from, 1% to maybe 15 or 20%. That seems like a very wide and uncertain range to give to anxious parents.
It is uncertain, and that's the toughest part of counseling for GM, because we usually can't sample the gonads to determine the exact proportion of mutated gametes. We have to rely on empirical data. Counselors often look at published studies for that specific disorder, what recurrence rates have been observed in other families confirmed or suspected to have GM.
So they might quote a general figure based on past experience.
Yes. For instance, for a condition where GM is well documented, a counselor might say something like, based on other families in your situation, the observed risk for recurrence seems to be roughly in the range of 5 to 10% for each future pregnancy. They acknowledge the uncertainty, but provide the best available estimate. The key message is, the risk is not negligible, it's significantly elevated.
So the family has to proceed assuming a tangible risk, even if the exact number is fuzzy.
Correct. But importantly, knowing his GM provides crucial, actionable information. The risk, whatever its exact percentage, is stable for all future pregnancies. This opens the door to reproductive options that wouldn't typically be considered necessary after a single de novo event.
Right. What specific options become relevant once GM is on the table?
The two main strategies are prenatal diagnosis, PND, and preimplantation genetic diagnosis, PGD.
Okay, tell me about this, PND.
With PND, testing is done during an ongoing pregnancy. This usually involves procedures like amniocentesis, sampling amniotic fluid, or chorionic villus sampling, CVS, sampling placental tissue, typically in the late first or second trimester. The fetal DNA is analyzed for the specific mutation known to be segregating in the family due to the parent's GM.
So PND confirms if this particular fetus has inherited the mutation.
Yes. It gives the parents definitive information about the current pregnancy, allowing them to make informed decisions based on the results.
And PGD, that sounds like it happens earlier.
PGD is even more proactive, yes.
Hmm.
It stands for pre-implantation genetic diagnosis, and it's used together with in vitro fertilization.
IVF. Oh, IVF.
Right. Eggs are retrieved from the mother and fertilized with the father's sperm in the lab. The resulting embryos develop for a few days. Then, crucially, one or a few cells are carefully removed from each embryo and tested for the specific mutation carried in the parent's germline mosaic.
You test the embryos before they're transferred to the uterus?
Exactly. Only the embryos that are confirmed not to carry the mutation are selected for transfer back into the mother's uterus to attempt pregnancy.
Wow. So PGD essentially allows the parents to filter out the risk before the pregnancy even begins.
That's the goal. For a parent known or strongly suspected to have GM with, say, a 10% recurrence risk, PGD offers a way to dramatically reduce the chance of having another affected child, ideally bringing it close to zero for that specific condition.
So let's loop back to the osteogenesis imperfecta example. A couple has had one child with severe OI, then learns through recurrence or maybe deep sequencing that one parent has GM. PGD becomes a powerful option.
A very powerful option. Facing the prospect of potentially having another child with severe brittle bones is incredibly daunting. The GM diagnosis, while difficult news, provides the rationale for considering PGD IVF. It allows them to choose, with high confidence, to implant only embryos tested and found free of the specific OI mutation. It moves them from a position of fear and uncertainty towards proactive management of the risk.
That knowledge, however it's gained, is key for informed family planning. It changes everything.
It really does. #tag3. Conclusion. So, to wrap this up, Gonadal Mosaicism really solves that genetic paradox we started with. How healthy parents can have multiple children with the same dominant disorder. The key things to remember are its origins, a postzygotic mutation happening very early in the parent's own development, and its location being strictly confined to the germ cell line, the cells making sperm or eggs.
Meaning the parent's body and health remain untouched, masking the risk.
Precisely.
So what's the big takeaway here? For clinicians, for students, for anyone listening, it seems like a huge reminder that our genes aren't always a single, uniform story throughout our bodies.
That's exactly it. And the most critical point, perhaps, especially if you're in a clinical setting, is this. If you see what looks like a de novo autosomal dominant disorder, but then it recurs in a sibling, you have to stop. You have to look past the standard rules. Gonadal mosaicism should jump right to the top of your list as the likely explanation.
Don't just assume lightning struck twice. Assume there's a hidden source.
Assume there might be a hidden stable source. It forces us to reconsider what carrier state means. You can be phenotypically normal, test negative on standard blood tests, yet still carry and transmit a significant recurring risk.
It blurs the line between your own personal genetic health and the genetic legacy you might pass on.
It really does. And that brings us to a final, maybe slightly unsettling thought to leave you with.
Okay, let's hear it.
If these postzygotic errors happening right after fertilization can set up stable, hidden mutation reservoirs in our germlines, reservoirs completely invisible to routine checks, and only revealed through reproduction, well, what other silent variations might be lurking? How many of us might be low-level germline mosaics for something? Something we just haven't had the opportunity to transmit or see recur yet.
So we might all carry hidden low-level risks that subtly change the genetic dice roll for the next generation without any outward sign.
It's a possibility. It certainly makes you think about the definition of genetically normal or healthy at a much deeper, more complex level, doesn't it? It suggests there might be more hidden genetic diversity within individuals than we usually appreciate.